Hi, I'm Lucas, and these are my observations for my decomposition lab. So, first of all, my hypothesis was if night crawlers are added to the container, then the trash items in that container will decompose more than the trash items in the control container. For my predictions, I predicted that the items in the experimental container will have decomposed more by the end of the experiment, and I guess at a higher rate of decomposition. So my experiment, like most others, consisted of two different bins. One of them is a control bin, one of them is an experimental bin. I have 10 pieces of trash in each bin. Um, there is 10 different types of trash and two of each. So I essentially have the same things in each bin. One bin obviously has night crawlers in them. There's about an inch and a half maybe of dirt in the bottom. I put 20 milliliters of water in each one, and they both stayed in the same location at about 68 to 71 degrees Fahrenheit, depending on what our thermostat was at. The experiment was held over 14 weeks, and I took observations every Sunday to keep it the same each week. My control variables would have been the amount of water and dirt, temperature, location, amount of light, and the amount of holes that were in the lid of each container were all the same. My independent variables were the night crawlers because they were in there changing the rate of decomposition or total decomposition, which is my dependent variable because that would change based on the night crawlers being added. So for my analysis of the data, first we can look at the control bin. I have a table and a graph. This table is showing by numbers really what exactly the mass lost of each item was, and this graph is just to put it into a little bit more perspective so that when we look at the experimental graphs, it'll look a little bit more clearer to you. So I took pictures, wrote descriptions, I took the mass and the mass change of each item, and that's about it. They both started off very similar. Not much was changing very drastically in the beginning, but as the weeks went on, I could definitely see that the experimental bin was doing some more work. So in order to really see what the difference was, we have to look at the other one. But as you can see, in both bins, the ham, cheese, yard waste, and the pineapple and watermelon fully decomposed by the end of the experiment. I will say, though, that in the experimental group, the rate of decomposition was much higher for those things, and they disappeared a lot quicker. The worms also made a lot more changes. They made less mold, but higher decomposition. And you could tell because the papers and stuff like that, they were changing colors and withering down very quickly. So here on the experimental table, we can see that the biodegradable and the paper's mass loss was much higher than that in the control group. The plastic and wood actually was also a little bit higher, but not very much, and the metal did not change much. So the nightcrawler's presence definitely did affect the rate of decomposition and the total decomposition by the end of the experiment. In conclusion, we look at the data and we can obviously see that the presence of the night crawlers definitely did affect the decomposition in the experimental bin. The things in the control bin didn't really change colors as much. They took a little bit longer to disappear, as you can see in the pictures. Um, and there was just a lot more damage in the experimental bin. The data also proves that by showing that the mass lost was much higher in the experimental bin when it comes to the biodegradable and the paper. That's where we really see the changes. So right here are the final pictures that I have of the experiment on week 14. As you can see, in the experimental bin, everything is much darker than it is in the bin. The control bin things look still a little bit intact when it comes to the metal and plastic and the wood and paper and biodegradable. But in the experimental bin, the paper and biodegradable are very withered and brown. You can see the wood has lost a little bit of its paint, and things just look a little bit dirty in there. So by the end of the experiment, my predictions and my hypothesis were both supported. To finish, I just have some further questions that can be raised from this experiment to keep experimenting with it and find more out about this topic. First of all, what worms can speed up the rate of decomposition? Would red regular worms, which are a very popular composting worm, work better in this situation? What environmental conditions allow for more efficient decomposition? So, if I were to change the amount of water and change the temperature and the amount of sunlight and ventilation that that bin is getting, would that rate of decomposition change? Would it be faster or slower? And finally, what can this data aid in composting research? Because that is mainly what we're looking at here is researching decomposition. We can really look at what can be composted, what can be turned into useful fertilizer in farms, and what can we do to make that process more efficient. And that is all I have for today. Thank you.